Okay. Now, of course, you will study everything.
Mhm.
These are just questions that seem tricky.
Yeah.
Here we have March 24, paper 2, and they were asking a question. Excuse me, here. No, these are the answers. I don't want the answers. Yeah. Using the information in the table, regardless of what the table is, explain the effect on the final protein structure of a substitution of the nucleotide at position 3. I added this question so that we revise the mutations, the type of mutations, and specifically the substitution mutation. What happens in a substitution mutation, Farouk?
A base is changed with another base. For example, T is changed with C. Or
Very good. So this substitution mutation, you have one base, it will be replaced by another base. Now, what are the effects of this substitution mutation?
it only changes one amino acid or one base triplet.
Okay, you are correct. One base triplet is changed. Now, this base triplet that changes can lead to what? Can actually lead to three things.
It can change the protein produced.
Yes, it can change the protein produced. Okay, and when it does change the amino acid, the base sequence, and finally the amino acid sequence and the protein that is produced will be different. Then it's going to be a functional or a non-functional protein. It's going to be the protein that we want or a protein that we do not want.
It could be both. Can be a protein that wants, or the amino acid it might have the same code. Don't multi.
Yes, yes, when the base is changed, but still the same amino acid is produced, and the final protein, it's the same. It did not change. Which type of silent mutation is that? Because we have three types of silent mutation. Oh, I'm just told you the answer. The three types of mutation,
It's
three types of substitution mutation. You have in substitution mutation, one base will be substituted by another base. Sometimes this can cause a problem, and sometimes it will not cause a problem. You just said that sometimes the same amino acid is produced. When this substitution mutation results in the same amino acid and the same final protein, what type of substitution mutation is that?
side.
Silent. Okay, now can you give me a reason why, even though we changed the base and there was a mutation, but the final amino acid was the same?
Because the amino acids gonna have different codes, different base triplets that produce the same amino acid.
Okay, the base triplets changed, but the same amino acid is produced. You did not give me an answer. Even though the base triplet changed, a
Mm.
mutation did happen. How come the same amino acid was produced?
Because it's degenerate.
Exactly. This is the word that I was looking for. Now, what does degenerate mean?
it can have like multiple codes, multiple base triplets produce the same amino acid.
Exactly. Now, you know the answer. Just write it down. Be generous in your answer. I always tell you that you're always summarizing your answers. Write all this information down. Now, when the substitution mutation happens, but in this case, we will produce a different protein. This is something good or bad?
Bad.
Bad. Okay. Either it will produce a different protein altogether, or it can lead to a premature stop and a shorter protein, which fine. In both cases, it will lead to a deformed protein that is non-functional. So either this substitution mutation will have no effect because, as you said, the base triplets they can change, but they still code for the same amino acid because base triplets are degenerate, or the substitution mutation will lead to a changed amino acid and a changed protein, either a defective protein or a premature protein. In both cases, it is undesirable. Okay. And a premature protein or a protein that is shorter than the original protein, it has a name, but you don't. Do you want to remember the names together?
Or
We
yeah,
have here silent, the silent substitution mutation. This means that then the base change will not lead to any change in the final amino acid and the final protein because the base triplets are degenerate. Nonsense mutation, this when the protein that is produced is shorter than the original protein, it will lead to a premature stop. and the missense mutation in case they ask you for those names. This is when the base substitution will lead to a different amino acid and a different protein that is non-functional.
Mhm.
Okay?
Yes.
Like we have an example that we studied, the sickle cell anemia. The original base sequence is GAG, GAG, and it is substituted A, the adenine is substituted to T, it will change to GTG, and it will give you a different amino acid. This amino acid, how is it different from the original amino acid in sickle cell anemia?
It changes the shape of the red blood cell.
Yes, it doesn't carry enough oxygen. It's not as efficient in carrying oxygen. It changes the shape of the red blood cell. Also, we took more information, Farouk. The original amino acid has a name, and the mutated amino acid has a different name and different characteristics. What's the original amino acid?
Uh, Glycine.
No, glutamic acid.
Glutamic acid.
And the mutated amino acid is valine
Okay.
or valine. And
Miss,
the
what
huh?
are the subcodons؟ مش في subcodons لازم أحفظهم؟
No, the term stop codon is enough. You don't have to know the name of the stop codon. The stop codon is the codon on the messenger RNA that will end the translation. It will tell the ribosome that translation finished. We keep on producing the amino acids and the polypeptide chain until we reach a stop codon in translation, and then the translation will stop.
مش لازم أحفظ certain subcodons يعني. هو
Oh. We usually give it to you in the question.
okay, thank you.
Okay. Uh, Also going back to the hemoglobin, the mutation in hemoglobin, we change again the glutamic acid into valine. The glutamic acid is hydrophilic, it is polar, it is water soluble, and the valine is hydrophobic and it is non soluble. And this is why it makes the hemoglobin sticky and sickle in shape and not efficient in carrying oxygen and not as soluble as the healthy hemoglobin. Okay, don't forget that. Good. And this mutation happens in the beta globin polypeptide chain of the hemoglobin. You know the structure of the hemoglobin. If you were studying biological molecules, it is made up of two polypeptide chains bonded together. You have two alpha globin polypeptide chains and two beta globin subunits. And the mutation happens in the beta globin, not in the alpha globin.
Mhm.
Okay. Good.
Yes.
Here, it's all written in your notes. The glutamic acid is soluble or polar, is now valine, that is insoluble or non-polar. Okay, going back to the question, we are revising everything.
Yes.
Here, this also March 24, use the information in the table, state and explain the effect on the final protein structure of a deletion. Now, the other type of mutation, here we deleted one base.
Mhm.
What's the difference between a deletion mutation and a substitution mutation?
In the deletion, there will be a knock-on effect. It will affect all the amino acids after that.
Yes. Okay, it will have a knock-on effect, and there is a name. These type of mutations, they're called what? Frame
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 shift.
frame shifts. Yeah.
Frame shift. Why is it called frame shift? Because when you remove a base, all the other bases will move to the left. They will all be shifted to the left, and the mutation is going to affect the sequence of bases from the point of mutation onwards, and all the amino acids that will come after the mutation will be altered, and the final protein will be altered. This has a drastic effect. Okay, so we revised all types of mutation in those two questions. Let me see here. Explain how the ability of the immune system to resist the damaging effect of a pathogen is affected by the destruction of the T helper cells. The lesson immunity is very important. Now,
Yes.
the T helper cells are destroyed because of an infection, because of HIV. How will this affect your immune response?
Less cytokine cytokines are produced, so less B lymphocytes and T killer cells are stimulated. So the B
Yes,
lymphocytes won't differentiate into plasma cells and memory cells, so less antibodies are produced from the plasma cells.
correct. Okay. Everything that comes after the release of the cytokines. Cytokines activate B lymphocytes, T lymphocytes, more macrophages. All this is going to drop. When there is a decrease in the B lymphocytes, you just said it, no antibodies will be produced and no memory cells. Don't forget. Okay. And when there is a decrease in the T lymphocytes, what's the function of the T lymphocytes?
Right. They secrete cytokines.
They don't secrete anything. B lymphocytes secrete antibodies.
T.
T lymphocytes, do they secrete antibodies?
No, they secrete. T lymphocyte.
Do they?
T helper. Oh,
Uh,
sorry. Confused. They kill the pathogen.
yes. T
Right.
lymphocytes
Sorry. Yeah.
are subdivided, or the types of T lymphocytes that we studied are the T helper and the T killer, or the T cytotoxic. The T helper, you already said it, it produces the cytokines. And the T killer, these directly kill cells, directly kill infected cells. They do not produce any antibodies.
Do I
Okay.
need to
And
know how?
sometimes they kill, excuse me?
Do I need to know how they kill them?
Excuse me? I can't hear you.
Do I need to know how they kill the pathogens?
Yes, of course.
Okay.
We said it many times. The T lymphocytes, there's nothing hard about it. They don't produce antibodies. They just directly kill infected cells. They're part of the cell-mediated
Yeah.
immunity.
I mean,
Okay.
how they kill them? Do I need to know?
They I saw it in the mark scheme, and you will find it in your notes that they produce a chemical. It's called the perforin or hydrogen peroxide that creates holes in the cell wall of bacteria, for example. So they will die. They
So
will
it's
take
like,
in water by osmosis and, excuse me?
so it's like penicillin. It's basically the same thing.
Yes.
Okay.
Yes, they create holes, and this is one of their methods of destruction. They create holes in the cell wall of pathogens and they die. Okay. And also, there will be no clonal selection, no clonal expansion, no memory cell production. All this will be affected. Outline how anti monoclonal antibodies can be synthesized in the hybridoma method. Now, outline the steps of the hybridoma method, Farouk.
Okay. You're gonna inject a rat with a pathogen or an antigen
Yes.
of the CCR5. Okay.
Okay.
That would stimulate the immune response.
Yes.
So the B lymphocytes will differentiate into plasma cells and memory cells. We're gonna extract the
Yes.
plasma cells from the spleen,
Excellent.
and then we're gonna fuse them with the myeloma cells so they can
Yes.
have uncontrolled mitosis. And then we're
And
gonna
when they
screen
fuse, they create a cell called what?
uh, hybridoma.
Yes.
And using a fusible such as a small electric shock, uh,
Good.
and then we screen them and humanize them.
Good. Okay. After we screen and humanize them, and
Mhm.
then they take the green light. Okay, they are screened now and they're producing the antibody that we want, and they're humanized so that they don't cause allergic reactions. They will then be allowed to clone. We will put it in a fermenter so that they multiply and multiply, they divide by mitosis and give us the monoclonal antibody that will be used in which type of immunity?
Uh, Artificial passive.
Good. Excellent. Okay. Explain how the primary transcript is modified before translation. Which step are they talking about here in protein synthesis?
Splicing.
Yes,
We
this is
remove the
splicing. What happens in splicing?
we remove the non-coding introns and join the coding exons.
Very good. Okay. And fuse the exons. Now, they have been asking about the introns and the exons lately a lot. All these are recent questions from recent exams. Describe the role of the hydrogen bonding in the movement of water through the xylem. Now, discuss the movement of the water up the xylem.
So they move up by adhesion, which is hydrogen bonds between the water molecules and and the walls of the xylem, and by
Excellent.
cohesion, which is hydrogen bonds between similar water molecules.
Very good.
And
Now, can you draw it? Can you draw like the cohesion between the water molecules? Let
yeah,
me help
between
you draw it. Uh, I
between
have
the
a water
hydrogen
molecule.
and the oxygen
Between the
and
what?
gas is between the
Yes.
oxygen and hydrogen and gas lines.
Perfect. Now, the oxygen, next to the oxygen, what charge do I put?
Positive.
Again,
The
next to
hyd-
the oxygen.
All oxygens are negative.
Negative. And do I do it like this or there is a sign?
There's a sign because it's
Yes,
uh,
electronegative because it's slightly negative charge due to the unequal sharing of electrons. And the hydrogen positive or negative?
positive,
Okay,
electropositive.
electropositive. And then here we have another water molecule. Also, this is electronegative. This is electropositive. And then the hydrogen, do I make a straight line or a dashed line?
Dashed because it's
Dashed line.
electron.
Okay. Okay, very good. This is the hydrogen bond. Of course, if you noticed in any mark scheme, they tell you that you can use annotated diagrams. The hydrogen bond here, this is going to give you a grade. You'll discuss it. Okay, we talked about the hydrogen bonds. Talk to me about another factor that also pulls the water up the xylem.
The transpiration pull at the
Yes.
leaves, water vapor leaves by transpiration, creating a gradient, so the water moves up down the concentration gradient.
Okay, down a concentration gradient. In other words, from high hydrostatic pressure to low hydrostatic pressure. Where is the high hydrostatic pressure?
Down
The
by the
soil or the leaves?
by the roots.
The soil has high hydrostatic pressure
Yeah,
or at the leaves?
the soil.
The soil. Yes, you said that at the leaves there is transpiration. So if this is the tree and these are the leaves, I have water vapor here evaporating, diffusing out of the stomata and transpiration. So I have low hydrostatic up there at the leaves. And in the soil, I have a water supply. Either I'm watering the plant or there is rain. So I have here high hydrostatic pressure. And you just said it that the water is going to move down a concentration gradient from high pressure to low pressure. This is going to create a transpiration pull, pulling the water molecules. Now, what is my question? There is a key term, a keyword that we mention here due to the movement of the water as one unit inside the xylem. Starts with the letter M.
And uh,
Mass.
mass flow.
Yes, always mention this term when you are talking about the movement of water in the xylem and the movement of sucrose and amino acids in the phloem. Okay? Mass flow. Even the movement of the blood in the blood vessels. Always liquids in a tube, like a blood vessel, xylem, phloem, they move from high pressure to low pressure.
Yes.
Okay? This is the mass flow or transpiration pull in the xylem for the reasons we said cohesion, adhesion, and transpiration pull. And you explained it well. Very good. Now, draw a diagram to show the arrangement of phospholipid molecules. You need to know how to practice sketching the cell membrane. Can you help me draw it, or can you draw if you can?
Uh, can I? I don't think I can. I can describe it. There is a circle and then two tails.
Okay.
The circle is onto the outside, that's the phosphate head, and then the fatty acid tails are under it, and then there's another
Yes.
one under it, but the fatty acid tails are facing each other.
Okay, that's it. This is all what I will draw.
Uh, that's the phospholipid molecules, but then you can draw
Correct.
also the channel proteins and
Yes, I can draw. Like you will draw at least five. Okay. And what are the structures that are embedded other than proteins? Like this is if a carbohydrate chain is attached to the protein, then this is called what?
a cell signaling molecule.
It is a cell signaling molecule, or it can act as a cell signaling molecule, but it is called a glycoprotein. Glycoprotein.
Okay.
Yes, it does function as a cell signaling molecule, or for cell to cell adhesion, or cell signaling, or communication, or as a molecular marker, an antigen. Good. Glyco means carbohydrate. Carbohydrate attached to a protein. And if this carbohydrate is attached to a lipid, it's called glycolipid.
Okay.
Okay. What other structure that is also embedded within the phospholipids that is really important for the stability of the phospholipid?
That's true.
Cholesterol. I can draw here a structure. Okay. And label, of course, label all that. Or you can draw a channel protein, carrier protein. Okay. You have several types of proteins. You have the glycoprotein, you have the channel protein for facilitated diffusion, and you have the carrier protein that has a specific shape like this. Okay, for a specific, and they're both specific. What's the difference, Farouk, between a channel protein and a carrier protein?
Carrier protein is for active transport.
Correct. Okay. Now, when they change shape, what is the name of this change of shape? To
So
take in molecules against a concentration gradient.
uh,
Con,
conformational changes.
yes, conformational change. Good. Okay. Now,
See,
here they want you to draw. Yes, my dear?
ينفع حضرتك تشرحي لي بس السيل سيجنالينج عشان كذا مرة جاب سؤال
Very easy. Yes, very, very easy. Cell signaling, it's so easy. It starts with the release of a signal. This signal can be a hormone or a neurotransmitter. So we will give it a general name, the release of a ligand. This ligand can be a hormone or a neurotransmitter, whatever type of signal is being released. This ligand is going to then be received by a receptor.
Mhm.
That's why step number 2 is called reception. It will be received by, of course, a complementary receptor.
Yeah.
After it is received by a complementary receptor, it's going to start a series of chemical reactions or a series of enzyme cascade reactions. Enzyme 1 is activated, then enzyme 2, and so on and so on and so on. So step number 3, it's going to start a series of chemical reactions or a cascade of reactions. Sometimes it is described as activating a secondary messenger. They all mean the same thing. You don't
Mhm.
have to write all of them down. One of them is enough. Activate a secondary messenger. And then finally, it will lead to a final response
Okay.
or action. And of course, the cell that contains this complementary receptor is called the target cell or organ. If it
Okay.
doesn't have a complementary receptor, then it's not a target. Okay,
Okay.
Farouk?
Yes, thank you.
You're welcome. Now, here is a picture of a cell in metaphase, and they want you to draw the stage of metaphase. What will you draw in that stage?
The chromosomes, the sister chromosomes at the equator connected. There's
At
a
the equator.
centromere
Very
between
good.
megavolum
The
connected to
chromosome is made up of a single strand or two identical strands.
two identical strands.
Yes, so you will draw two identical strands held at the centromere. Okay, what else?
The spindle fibers,
Very good. Coming from where?
the uh,
Yeah, the centriole or
century,
centrosome,
yeah,
same
century,
thing. And the spindle fibers connected to the.
centromere.
Yes. So you will draw something like this, but don't forget to label. Okay, labeled diagram. Here is the chromosome, and it's nice to clarify and say it is made up of two genetically identical sister
Chromatin,
chromatids. And this is what is the central meeting point between both?
centromere.
Centromere, good. And what do I label this?
Spindle fibers.
Yes, spindle fibers. And what do I label this?
Centriole.
Centriole, good. That's it. Very good. And this is the equator or the metaphase plate.
Okay.
Perfect. You would because it's 3 marks. Like when you see a question, 3 marks and above, be generous in the question. Always write more. More is better than less. Okay.
Mhm.
Now, outline the changes that occur to the structure and behavior of a chromosome from the start of the synthesis phase to the end of interphase and during the prophase.
Okay.
Now, I put this question so that we revise all the steps of mitosis. Tell me what happens in the um, interface. Let's
Okay.
talk about interface.
It's three parts. There's G1
Yes,
is S phase, and then G2. And G1
good.
is growth of the cell, and then
Yeah,
the S phase is the DNA replication by semi-conservative method, and then the G2
excellent.
is the growth of the organelles.
But something really important happens in G2. We check, we make sure that the semi-conservative replication of the DNA happens properly. Okay, we check for errors. So more growth, yes, you are right. And we also check for errors so that we don't start mitosis except when everything is nice and clear. So we check for errors.
Yeah.
Good. Now, what happens in mitosis? What are the steps of mitosis?
Prophase, metaphase, anaphase, and telophase. In
Good.
prophase, the chromosomes condense, they're visible.
Yes.
In the
This
meta,
is the early prophase. Okay, they condense. Correct. Condensation means what?
they they like become thicker.
Yes, correct. And then what disappears in prophase?
The nuclear membrane,
Very good. What else?
the uh, uh, the, the
The
nucleolus,
center of the yes, the nucleolus also disappears. And what appears?
what the chromosomes, concealing.
We said that they become shorter and thicker and they condense. And what is going to move to opposite poles of the cell and start to
Yes,
produce the spindle fibers? The
centrioles.
Yes, they start moving to opposite poles of the cell. One will move here, one and move here, and they start producing these spindle fibers. Good. Now, what happens in the second phase? You said metaphase.
Yes, metaphase. The chromosomes, the sister chromatids, are aligned at the equator, connected by spindle fibers that are connected to the centromere.
Good. What happens in anaphase?
The centromere splits in half, so each chromatid is pulled to opposite poles of the cell by the spindle fibers.
Excellent, excellent. Don't forget to mention that the spindle fibers, they will contract, they will become shorter. Good. What happens in the next phase? What's the name of the next phase
Telophase.
anyway? Hmm?
Telophase.
Yes, what happens in telophase?
Cytokinesis. The cell splits into two, divides into two.
Yes. And do you remember the difference between no, 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 wait. So this is cytokinesis. What happens in
Oh,
telophase?
so yeah,
It's different.
yeah, yes. The cell membrane is pinched.
This is in cytokinesis. Telophase, what will reform?
The nucleolus and the nuclear membrane.
Yes, they will reform. And then in cytokinesis, the cell membrane will pinch off. This is in the case of the animal cells. But how does cytokinesis happen in plant cells?
There's something is formed. It's I forgot his name.
What do you mean something? Vesicles, they will arrive from the Golgi body and they will deposit
Uh,
cellulose. Okay, they will deposit a central plate. This central plate will then be filled with cellulose, and then the central plate will join to form a cell wall.
yes,
Cytokinesis in animal cells and plant cells are different. Revise them. Okay. In the
yes,
animal cells, the cell membrane will pinch off and form a cleavage furrow until it separates. But in a plant cell, we have vesicles coming from the Golgi body, and they will lie in the middle, and then they will deposit cellulose, and these plates will fuse together to form a hard cell wall that will separate the two plant cells. Okay.
okay.
Now, describe the mode of action of the Kupffer cell in removing and breaking down a damaged red blood cell. In other words, we want to revise the steps of phagocytosis.
Phagocytosis. The macrophage or the phagocyte engulfs the substance, forms a phagosome around it, and then
Okay.
the lysosome fuses with the phagosome, forming a phagolysosome. And they release hydraulic enzymes such as uh, pro- protease and,
Yes.
and nuclease to
Yes.
break
Yes.
down
Stop here. The nuclease. This nuclease enzyme, what does it break down?
the nucleus.
Yeah. No, it doesn't break down the nucleus. It breaks down a bond that is found in the nucleus, the phosphodiester bond.
Okay.
Okay.
Yes.
And yes, you need to memorize some enzymes. And there is also another enzyme. It's called the lysosome or lysozyme. Where
Yes.
is this question? And what does this break down?
Uh, I don't know this way.
Here, I added it here in the mark scheme because, and it is in red. Look here. Examples of hydrolytic enzymes found in lysosomes. Carbohydrates, protease, lipase, nuclease, and lysozyme. Nuclease and lysozyme are the two enzymes that usually confuses students. The nuclease, okay, an enzyme that breaks down the phosphodiester bonds. And the lysozyme, it breaks down the peptidoglycan in bacterial cell walls.
So,
Okay?
can I still say the lysozyme here, even though we're
The
talking about
lysozyme, what?
cell? Can I still say lysozyme here, even though we're talking about the red blood cell?
We're talking about phagocytosis.
Oh, but this
This
is about
phagocyte,
red blood cell.
I'm talking about phagocytosis in general. If it
Okay,
is going to digest a red blood cell, no, of course
okay,
not.
so I
But I'm
should.
talking about phagocytosis in general, okay? First, it will be attracted, and then it's going to bind to the pathogen. Either it will engulf it directly, or it's going to bind to a complementary what that will mark the pathogen. Sometimes the pathogen is marked by an antibody, yes?
Yeah,
Okay. Now, this phagocyte, how does it bind to this antibody? What does it have to bind to this antibody?
complementary receptor.
Yes. So you have to mention all the steps. The
Mm.
phagocyte is going to be attracted to the foreign antigen, either due to the release of histamine or because of an antigen presenting cell, but it's going to be attracted. The attraction is called chemotaxis. And then it's going to engulf it either directly or it's going to attach to it because it has a complementary receptor and the pathogen itself is marked by an antibody. Regardless of how it will be attached, but it will be attached to it, it will engulf it and put it in a phagosome, like you said. And then the lysosome will come and release the hydrolytic enzymes, and the lysosome will fuse with the phagosome to produce a phagolysosome. And these enzymes, you give one or two examples until it will rupture. And if you're talking about neutrophils, dead neutrophils, they form a pus. And macrophages, they create antigen presenting cells. Did you forget this, Farouk?
No, I remember.
Okay, good. Fine.
Immunity is one
Explain
of
how, excuse
those.
me?
Immunity is one of the chapters اللي كنتها على طول حفظتها على طول.
Yes, immunity, the structure of the antibody, the making of monoclonal antibody, the types of the immunes, and they're easy, by the way. It's an easy lesson. We revised it and explained it a lot of times until I think now you digested the lesson really well. And phagocytosis is so easy. It's good that they repeat something that is so easy.
Yeah,
And I want you to focus also on gas exchange and cell membrane. Did you look at the modifications that I sent?
not yet. أنا بقالي فترة قاعد بس بحل بس بيبرز عشان أتعود على الأسئلة، بس دلوقتي بقى قاعد براجع الشابترز اللي أنا ضعيف فيها. The two chapters
Okay.
that I read most is biological molecules and cell membrane.
The cell membrane is easy, very
Yeah, it
easy,
says
but
that.
it's frequent. Like they frequently ask questions.
Mm.
Okay, now let's look at this question. Explain how blood flow through the alveolar capillaries help to maintain steep diffusion gradients for gas exchange.
Okay, the blood arrives in the capillaries with a
Mm,
low concentration of oxygen, so the oxygen
yes,
can diffuse in down concentration gradient and has a high concentration of carbon dioxide, so that the carbon dioxide can diffuse out into the alveoli.
very good. Now this question, surprisingly, it's common. Okay, and this is going to create a gradient. Also, something else creates a gradient: hyperventilation, a high supply of oxygen and low supply of CO2 in the inhaled air. This also creates a concentration gradient and helps in gas exchange. Explain the role of the elastic fibers in the alveoli.
They allow the alveoli to uh, expand more, so it can allow more air in, and it
Okay,
allows
I want you're not wrong, but there is a term that we use with the elastic fibers: stretch and
and recoil.
yes. Okay, these are the alveoli and the elastic fibers. They are running around the alveoli. They allow the alveoli to stretch and recoil. Now, what happens when these elastic fibers are broken down in infection or you're a heavy smoker? What happens?
The alveoli can no longer stretch, so it bursts, or if it doesn't
Yes.
burst, it cannot recoil to remove all the carbon dioxide or the excess air out.
Very good. So the air will be trapped in the alveoli. The alveoli will be larger and it will have trapped air, and a lot of them will
So also
rupture. Now, when a lot of alveoli rupture, yes, I can't
decreases
hear you. What did you say?
air. When they rupture, it decreases the surface area of the lungs, so
Excellent.
slower. Yeah, thank you.
Yes. Okay. Less surface area to volume ratio. Perfect. This is the problem with infection and the problem with bursting of the alveoli, decrease in the number of alveoli, and less surface area to volume ratio. Of course, poor oxygenation, less oxygenation of blood, etc., 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 emphysema, and difficulty in breathing. Okay.
Yes.
Now here they're talking about the structure of the elastic arteries. Can you remind me of the structure of the artery in three walls, and specifically elastic arteries?
We have the tunica intima, and then the
Very good.
tunica media,
Yes.
and then the tunica externa.
Yes. Now, can you tell me the tunica intima? How do I draw it? It's made up of what?
It's made up of elastic fibers.
The tunica intima, the innermost layer, not the tunica media, the tunica intima.
Oh, sorry. It's made of single cell endothelium cell.
Yes, endothelial cells.
Yeah,
Now, these endothelial cells, they are tall and columnar, or they are flat and cuboidal and single layer. Describe them.
they're they're single layer.
Yes, there are cuboidal, a single layer of cells on a basement membrane. Now, what is their function?
Uh, uh, to allow for
Yeah.
fast feeding.
With the rough, they allow the smooth flow of blood with no friction.
Oh,
Just a
okay.
single layer of flattened cells, okay? Cuboidal or flattened cells. They are smooth, okay? To reduce friction. Don't forget. Now,
Yes,
discuss the second layer. What is the second layer, Farouk?
the tunica intima media. Sorry, has
Yes, the tunica media. Now, stop here. What does the tunica media contain?
elastic fibers, collagen, and
Mainly, y- yes, it does. It contains all the three tissues, but in this type of artery, mainly which tissue will be abundant in the tunica media?
the elastic fibers
Correct. And what is the function of the elastic fibers here?
allow the arteries to stretch so they don't burst from the high
Yes.
pressure blood.
Excellent. Okay, so that they don't burst. Now, let's talk about a normal artery that contains elastic fibers and smooth muscles. What is the function of the smooth muscle?
When it contracts, makes the artery smaller. Decrease is makes the lumen smaller.
Yes. So any muscle will contract and relax. Any muscle that contracts and relax, we say its function, it will control the diameter of
Yes,
the blood vessel. It will contract to make the lumen smaller, like you said, and it will relax to make the lumen wider. Okay,
yes,
it will control the amount of blood flowing inside the artery. And the elastic fiber, like you said, so that it doesn't burst and it can stretch when blood is flowing inside it at high pressure. Now, talk to me about the last layer.
and the tunica externa it just has
Yes. Mainly what?
collagen.
Yes. And what is the function of the collagen?
It prevents it from bursting.
Yes, it does prevent it from bursting and provide it with support and strength, and it helps it to attach to the adjacent tissues. Okay,
Okay.
good. Okay, and here there's a space for a diagram. So this question wants you to discuss the structure and they are encourage you to draw a diagram. That's why I tell you while you're drawing anything, draw it. While you're studying anything, draw it.
Yes.
Draw an artery, draw a vein, draw. Drawing helps you remember and they accept drawings. They encourage drawings in your answer. Outline the events that occur in the cell of P. falciparum to form the daughter cells shown in figure 1.1. Okay, now here this discusses mitosis. So we already answered this question.
Yeah.
Okay, so all the steps of mitosis, I had this question so that we revise it. Explain how antibodies will reduce the spread of malarial pathogen through the bloodstream. Remind me, what is the mode of action of antibodies? How do
I know
antibodies
about
work? Yeah. Okay. I know ball. What is I for?
immobilization.
Yes. Now,
They
what would you say here?
they attach the flagellum to stop the bacteria from moving.
Good. And what about N?
Neutralization.
Yes, what do you say here?
They neutralize the pathogen.
Good, the toxins. Yeah. And what about O?
Opn, opno, optimization.
Opsonization. Good. And what will you say here?
I don't know to be honest.
Come on, here they will mark the pathogen so that it make it easier for the neutrophils to identify it and make it easier for phagocytosis. This is one of the most important mode of actions for antibodies. They will mark the pathogen,
Yes.
okay, to encourage phagocytosis so that the phagocytes can easily identify the pathogen. This is the pathogen. It is marked by an antibody. This is opsonization. Okay,
Yes.
so that the phagocyte can come and see it. It will only bind with it when the antibody is marking it because it has a complementary receptor. Here,
Yes.
okay, what is B?
Uh, blocking,
Yes. What is A?
agglutination, agglutination,
Agglutination, which is the same as immobilization. It will stick them together to prevent them from spreading. And what is L?
lysosomes,
Lysis. It will make it rupture. Now, when you take ready-made antibodies, this is an example of which type of immunity?
uh, art, artificial passive.
Good, perfect. Discuss the factors that contribute to the sources to the success of a vaccination program. This is a 5 mark question and actually very easy. What will make a vaccination program successful?
If the bacteria is not resistant,
You mean it's not resistant or it doesn't mutate? It is
and
stable.
yeah, it doesn't mutate.
Yes, does not mutate. Now,
Yeah,
how do we
enough
describe that? If it mutates, no antigenic what?
uh, or antigenic variation.
Yes, no antigenic variation. Good. No
Uh,
antigenic concealment, it doesn't hide. No antigenic shift, it doesn't change throughout the life cycle of the pathogen. It doesn't mutate is enough. Give me another reason for, or several reasons for the success of a vaccination program.
if most of the population is vaccinated, that will cause
Yes.
good immunity, so decrease in
Good.
spread.
When we vaccinate nearly 99% of the population, this has a name. It starts with an
Herd
H.
immunity, herd immunity,
Yes,
herd
perfect. We can achieve
immunity.
herd immunity. Another reason. So it's out of
Uh,
5, we want 6 reasons.
if the vaccine is free,
It's free? You mean it's not expensive? Okay.
yeah.
What else?
I mean, like free to the population. They don't have to pay ticket.
Okay. So the
Uh,
government can afford it, yeah? Because some
yes.
governments cannot afford it. Okay.
Uh,
Hmm. Do we need a booster dose or we don't? And does this affect the success of a vaccination program?
if you don't, it's more likely to be successful.
Yes. No booster dose. Now, which vaccination will be more successful? Using a live vaccine or a dead vaccine or a dead
A
pathogen
live
in the vaccine?
vaccine is
A live
better, but we
stronger. Of
can.
course, there are drawbacks, but a live vaccine is stronger. It will give you a stronger immune response.
But the dead
What else?
vaccine doesn't cause the disease doesn't cause infection, and it can be given to people with weak immunity, right?
Yes, yes, of course.
Yeah.
Each one of them has its own advantages and disadvantages. Um, Give me another reason for the success of a vaccination program. Talk about how the vaccine can be stored. Is it thermostable or not thermostable?
Yeah. If
Okay,
it's
this
if
question
it's
is common.
easy,
Excuse me?
if it's thermostable, it's easier to store.
Yes, of course. It's not going to
Yeah.
be destroyed by high temperature and it is easy to transport. So it is
Okay.
thermostable. The main reason for a success of a vaccination program that the pathogen doesn't mutate. It's stable. Okay. Um, This is one of the reasons that will make a vaccination program successful, and it can be freeze dried. It is thermostable. It is not expensive. We only need one dose. You don't need a booster dose. You're using a live vaccine. The government is very cooperative. We have the funding. We have the money to make the vaccine. People are convinced. Some because, like in the COVID, people were not convinced and they did not take the vaccine. So the vaccination program failed. Okay, people have to be accepting the vaccine so that we reach a level of herd immunity or 90% of the population or more vaccinated. Describe the events that occur when starch phosphorylase catalyzes the addition of a molecule of glucose to the end of an amylose molecule. Now, you were studying biological molecules.
Mhm.
Now, discuss how can we add glucose molecules to each other and creating a polymer.
Like uh, by condensation reaction.
Yes, but this is a question out of 4 marks. So you will discuss what is condensation, how it happens. Now, let me break it down for you. Amylose is made up of which monomer?
Uh, Alpha glucose.
Alpha glucose. Good. So we are talking here about the alpha glucose as a monomer. Now we want alpha glucose bonded with another alpha glucose, creating a bond,
Yeah.
releasing water, condensation reaction. That's it. Okay.
Okay.
Now you cannot write here, but I want you to remind me the alpha glucose is how many carbon in the help me draw it. I don't know how to draw it.
So it's a hexagon.
Yeah. One, two, three, four, five, six.
Top right we hot O.
Yes.
Okay, and then it's C on all the others. C on
Okay.
here.
Uh,
OH تحت on the first one.
this
Carbon
is
is
alpha glucose. Yes.
yeah.
Okay. So the alpha glucose, the hydrogen is above or below?
The hydrogen is the hydrogen is above, and the sorry, the hydroxide is under. OH is under. Sorry.
1 minute. It's alpha glucose. So
Yeah.
the hydrogen at carbon number one is above.
Yes. That's what I'm saying. And the
Okay. And the
OH
OH will be
under.
below. Yes.
Yeah.
Okay. Good. And then what about the second one?
And then the OH is under as well.
Yes.
And
And
then
we
the
alternate. It's easy. We alternate down, up, down. What about carbon number? This is carbon number five. Where's carbon number six?
there's like a line, and then CH2OH.
Very good. This is one alpha glucose, and we will draw another one next to it. Exactly the same. Okay. So here, this is the O you said carbon one, carbon two, carbon three, carbon four, carbon five, carbon six is outside the ring.
Yes. And then when
Alternate. Then we do what?
we remove the OH and one H, that will form water,
Perfect.
and then there will be a bond between the O and the C, between carbon
Yes.
one and carbon four, like so.
Perfect. This is alpha 1 to 4 glycosidic bond, water release, and this is a condensation reaction. Very good. I want to finish half of this sheet today. We're close. Now here, this is an oxygen dissociation curve, and the curve here, it moved down. When the curve moves down, this is a Bohr shift. What does this Bohr shift tell you?
There is a decrease. There's a more carbon dioxide.
Correct. There's a decrease in what first?
The affinity to oxygen, the hemoglobin affinity to oxygen.
Okay, there's a decrease in the affinity of hemoglobin. What does a decrease in the affinity of hemoglobin mean?
The hemoglobin will not want to bind to oxygen and will want to bond to carbon dioxide more, or to something else
Good.
more.
Perfect. The percentage saturation of hemoglobin with oxygen, which how well the hemoglobin binds with oxygen, it will decrease. Good. And you just said it, that this happens when there is more CO2. Perfect. And the opposite happens when the curve moves up, increase affinity, more saturation, etc., etc. So here we already described the effect of an increase in the concentration on the oxygen dissociation curve. Now this is protein synthesis. Name the enzyme labeled A. Where is A? A is here. It is creating the messenger RNA. So what is A?
RNA polymerase.
Excellent. RNA polymerase.
Yes.
Good. Name the bond that forms between the nucleotides and the primary transcript.
Phosphodiester.
Excellent. Phosphodiester. State the number of amino acids that are coded for by the sequence of nucleotides on the primary transcript. How do you know how many amino acids coded for? What do you count?
Uh, you, you count the bases that are like
Correct.
on the
We
left,
count the bases, and every three bases code for.
one amino acid.
Good. So we will count the number of bases on the DNA or on the messenger RNA.
Or on the ones that are being coded for, so the messenger RNA. Okay.
Correct. So
Yeah.
let's count together. This is three, or those are three, and then another three, and then another three, and then one, two, three, and then one, two, three, and then this is one. So we will, this is not, we are one, two, three, four, five amino acids.
Yes.
Okay. Use the information in the figure to explain why one of the strands of DNA is not transcribed. Now, this also question, I saw it a lot. Why only one strand is transcribed and the other strand is not transcribed?
Because the uh, only there is
The
one
strand, there is one what?
template strand.
Yes, you're right.
Okay,
The
and
strand that is transcribed is the template strand, correct? And the other strand is not the template. Now, why is the other strand not the template?
it remains in the nucleus.
No,
No,
both strands remain in the nucleus. I want to ask a question. Both
uh,
strands are complementary, yes, but do they have the same sequence of bases?
no, they don't have the same sequence of base.
They don't have the same sequence of bases. So if we transcribe the other strand, it will have a different sequence of bases. This will lead to a different sequence of amino acids and a different protein.
Yeah,
It is
okay.
as simple as that.
Uh,
Okay. And I sent you the mark scheme. Go look at the mark scheme. So we only have one template strand. And the reason why we have one a template strand, because the other strand has a different sequence of bases, different amino acids. And this is going to make us produce a different protein that we don't want. We want this strand because the sequence of bases in this strand will give us the protein that we want. It is as simple as that. I'll highlight this question for you. Okay. Outline the ways in which ions can move into a mitochondria. This is a very easy question because I wanted to revise all the methods of movement of ions in cell membrane. How can ions move into any
they can move
organelle?
to by active transport against the concentration gradient using energy by carrying
Yes.
proteins. They can move by facilitated diffusion through channel proteins. They can
Yes,
move
correct.
through co-transporter proteins.
Okay. Yes. You mentioned, actually, you summarized all of them. Very good. By facilitated diffusion, co-transported proteins. Good. Describe and explain the mechanism that is responsible for the movement of the phloem sap in the sieve tubes. Now, I want to revise this question and the two steps here in the loading of the sugar first, and then the movement of the sucrose inside the sieve tubes. I'm going to answer more than needed. Remind me, Farouk, how the sucrose is going to move from the leaf into the cell wall of the companion cell.
Okay, there's a hydrogen pump. The hydrogen pump will
Excellent.
pump hydrogen outside into the cell wall,
Good.
and then the
Let me
hydrogen
try to. So I'm drawing here the cell membrane, and this is the cell wall, and he said that there's a hydrogen pump. Perfect. What does the hydrogen pump do?
it will pump hydrogen ions outside against the
Correct.
gradient.
Okay, so I will have hydrogen ions outside until we reach what?
And the high concentration.
Yes, you're right. A high concentration of hydrogen ions in the cell wall.
Okay, and then
Okay,
they
this is the cell
back.
membrane. Yes, this and this, you're right. And you can describe it this way, or you can say that we will create a proton gradient. It's just more sophisticated. We will create a proton gradient, or what you said is perfectly fine. We will have a high concentration of hydrogen ions, which are positively charged in the cell wall. And then what comes next?
The hydrogen ions will move through the co-transporter proteins and transport the sucrose with them. And
Excellent.
the sucrose will move against the concentration gradient and will use the energy produced from the hydrogen ions moving down the concentration gradient by diffusion.
Good. It's called secondary active transport. And the sucrose is going to move, or it will be co-transported with the hydrogen ions. The hydrogen ions will move in down a concentration gradient here by facilitated diffusion, don't forget. And as the hydrogen ions are moving in, they will create or they will power the energy needed to transport the sucrose against the concentration gradient. That's why it's called co-transport. Good. Now, this is the first step, but this is not actually what they want. They want the movement of the assimilates within the phloem itself, from the source to the sink. What happens at the source in the phloem?
Okay, so
And then you said what you said is perfect. Now we have sucrose here in the cytoplasm of the companion cell. How does it enter into the phloem? Through the what?
through the plasmostomata,
Correct. And this sequence of events that you mentioned, which involves the hydrogen pump and the co-transporter protein, is the apoplastic or the symplastic pathway?
s- symplastic, right?
My
No,
dear, the first step, the one that involves the hydrogen ions and the cell wall and the co-transporter protein, is the apoplastic or the symplastic?
apoplastic because it's going through the cell wall.
Correct. Perfect. So here, the first step in the loading of the sugar is apoplastic, and here it involves energy. Active
Mhm.
transport, good. And then the sucrose is going to increase in concentration in the cytoplasm of the phloem of the companion cells and then move through the plasmodesmata into the phloem. Now, as it moves through the plasmodesmata into the phloem, this is apoplastic or symplastic?
That's symplastic.
Good. And this is active transport or diffusion?
Diffusion.
Perfect. Now, here inside the phloem, we have a high concentration of sucrose. How is this then transported to the sink or carried to the sink
So
within the phloem itself?
there is a KFE low water potential.
Yes.
Okay,
Okay, initially there is a low water potential because of the high concentration of sucrose, and then this is going to draw what out of the xylem?
water from the xylem into the phloem.
Good. Yes, by osmosis. And when the water moves out of the xylem into the phloem, this is going to increase the what?
Hydrostatic pressure.
Perfect. So at the source and within the phloem, I have high hydrostatic pressure or low hydrostatic pressure.
Now is high.
Yeah, good. Now
Uh,
talk to me about what happens in the phloem at the sink.
okay, at the sink, there will be unloading of sucrose,
Perfect.
so there will be low hydrostatic pressure.
Good. But you need to elaborate. The sucrose is going to move out of the phloem and into the sink. Now, when it goes out, what happens to the water potential initially?
Increases.
Correct. And when the water potential increases, this is going to make what? What happens then?
That will decrease the hydrostatic pressure.
Yes, why? Because water is going to move in or out of the phloem at the
It
sink.
move out of the phloem
Yes, water
into
is going to move out of the phloem by osmosis down a concentration gradient from the phloem back to the xylem.
okay,
So I have high hydrostatic pressure at the source and low hydrostatic pressure where?
at the sink.
At the sink.
Okay,
Now, what does this remind you of? Changes in
there's
hydrostatic pressure will lead to what?
the pressure gradient, so they will move down the concentration gradient by
What's the term? Starts
diffusion.
with M.
Uh,
Mass
mass flow,
flow.
mass
Mass flow.
flow,
Mass flow.
mass
And
flow.
this mass flow does not require energy.
Yes.
Okay. Now, this needs revision. This is important.
Mhm.
We have movement of materials in the xylem and in the phloem. Okay, the reasons are different, but the naming is the same. In the xylem, it's called mass flow, and the phloem is also called mass flow. In the xylem, I have here degenerated end walls, so I'm going to draw a hollow tube, and the mass flow or the transpiration pull in the xylem is the reason of cohesion, adhesion, and changes in hydrostatic pressure. Okay, and we explained cohesion and adhesion and the change in hydrostatic pressure. In the phloem, the story is different.
Mhm.
The naming is the same. I have here the phloem with sieve tube elements here. I have a companion cell and I have the source, which is the leaf. The leaf is going to do photosynthesis and produce sucrose. And then this sucrose will move into the phloem through two steps. Number one, it will move from the leaf into the companion cell by the hydrogen pump and the co-transporter protein. And then from the companion cell, it will move through the plasmodesmata into the phloem. Okay, now I have sucrose here in the phloem. This is going to make the phloem very concentrated. This is going to attract water from the adjacent xylem, and the water is going to increase the hydrostatic pressure at the phloem. Now, at the sink, sucrose is being unloaded. So, at the sink, the phloem will be very dilute because the sugar is going out. Because it's very dilute, the water will move out of the sink back to the xylem, and we will have low hydrostatic pressure at the sink, high hydrostatic pressure at the source, low hydrostatic pressure at the sink, change in hydrostatic gradient, movement of the assimilates, the sucrose and the amino acids in the phloem from the source to the sink by mass flow. Okay? So the phloem and the xylem, the movement of materials within the phloem and the xylem, there is one thing in common: change in hydrostatic pressure, mass flow. But the reasons are different.
Yeah,
Okay, Farouk?
yeah.
Okay. In the xylem, I have loss of water at the leaves by transpiration. I have high water content in the soil. I have low hydrostatic pressure at the leaves because of transpiration. I have high hydrostatic pressure in the soil because of the water supply. So I have changes in hydrostatic pressure leading to mass flow in the xylem and the movement of water from the soil up to the leaves. And it has another name called the transpiration pull. Okay. And the phloem also assimilates move within the phloem due to changes in hydrostatic pressure. But the reason why the hydrostatic pressure is high or low is different. We don't have transpiration here. We have loading and unloading of sucrose that will make the source of high hydrostatic pressure and the sink of low hydrostatic pressure. This part you should never, or you can never memorize it. You have to understand it. Is it clear?
Yes, it's clear.
Any
Thanks.
questions? Okay, good, good, good. So let's solve this question. Now, here we're talking about the cardiac cycle. Can you remind me what are the stages of the cardiac cycle?
Yes, there's atrial systole,
Yes.
then atrial diastole and ventricular systole, then ventricular diastole and dis-
Wait,
diastole.
wait, 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 wait. Atrial systole, both atria contract, and then what next?
The atrioventricular valve is open. The water moves into
This
the
is not
ventricle.
part of the atria. This is not part of the cardiac cycle. These are the events that happens within it. I'm talking about the cycle. Only three events.
Atrial
Atria contract,
systole,
and then what will contract?
the ventricles,
Yes, ventricular systole, and
yeah,
then
just diastole and everything.
yes, that's it. Okay. These
Yeah.
are the three events in the cardiac cycle. Now, the reasons or what happens, you will tell me also.
Mhm.
What will initiate a wave of stimulation or activation
Sino-
or a wave of excitation in the heart?
The
Is
sinoatrial
there
node.
a stimulus?
Uh,
Excuse me?
The sinoatrial node.
Yes, correct. And I want you to tell me, is there a stimulus or there is no stimulus?
No, it's myogenic.
Good, perfect. So I have the sinoatrial node. I have a wave of excitation. Call it wave of excitation, not an impulse. They like it this way. And then from the sinoatrial node, where does it go? What
Across the
does it do?
atria.
Yes, they will cause the
Atria.
atria to contract. So it will cause atrial systole.
Yes.
And then after the atria contracts, where does it go?
It goes in the down to the goes to the septum to the atrioventricular
Okay.
node, which delays
Excellent.
the impulse,
Atrioventricular node that will cause what? A slight
delay to the ventricles can fill up
delay.
with blood.
Excellent. Now, something else causes a delay, and I told you to add it.
The the bundle of his
No.
uh,
Something else helps in the delay. And I sent you the image and I told you add it, but you don't listen. What is it? There is a tissue, a fibrous tissue between the atria and the ventricles here, called the annulus fibrosus. You don't have to know its name, but there is a tissue here. So this is the sinoatrial node somewhere here. Look at my arrow because I cannot write here.
yeah,
Sinoatrial node is going to release a wave of excitation. It's going to cause both atria to contract. And then this wave of excitation is going to reach the atrioventricular node here in the septum of the heart that is going to cause a slight delay. And something else also contributes to this delay. Fibrous tissue, you see this blue fibrous tissue between the atria and the ventricles. This
yeah,
fibrous tissue, it has a name. You don't have to know its name. You can just describe it as non-conducting tissue because I saw it in the mark scheme. It's called annulus fibrosis. You don't want to know its name, it's fine. But not only the atrioventricular node causes a delay, the atrioventricular node plus the non-conducting tissue between the atria and the ventricles. The non-conducting tissue that also contributes to the delay of the wave of excitation from reaching the ventricles. After a small delay, okay, it's going to continue along its pathway. It's going to go to the bundle of His and then to the Purkinje fibers that divide into two branches that carry the wave of excitation to the base of the ventricle so that the ventricles contract from the base upwards. Did you know all that or you're hearing this for the first time?
no, I knew everything but
Okay.
other than the non-conducting fibers or
Just
the non-conducting.
non-conducting tissue.
Yeah,
I saw it in the mark scheme. Uh, Please pay attention to the things I sent because I did not get it from the book. It's not in the book. I got
yeah,
it from the mark scheme.
yeah.
Okay, because the mark scheme, when they mention something, I suspect that they will get it in the next year. So I always concentrate with the mark scheme actually more than the book.
Yeah.
Okay, so here, what will initiate the heartbeat? What structure in the heart will initiate the heartbeat?
The sinoatrial node,
Good. What will cause a delay?
the atrioventricular node,
What else?
the non-conducting tissue
Good. The non-conducting tissue found where?
between the atria and the ventricles.
Ventricles. Okay, and look here, I've got it from the mark scheme.
Mhm.
Atrioventricular node and the non-conducting tissue or alternative wording between the atria and the ventricles. Okay?
Yeah.
It's not
Okay.
the septum, it's the non-conducting tissue. So these small details, we want to stress on them. What will transmit a wave of depolarization through the muscles of the ventricles?
The Purkinje tissue,
Yes. And what will close when the left ventricle contract?
the left atrioventricular node, or the
Left
bicuspid.
atrioventricular node. Okay.
Okay, I have to run
Okay.
upstairs.
You have a class? Okay. I'm happy we actually finished half the sheet. We can continue tomorrow.
Okay, thank you.
Okay. You're welcome. Bye.
Bye.